Dear students, up till now we have discussed that how Beadle and Tatum they worked on Neurospora spores, which is pink bread mold. Now the next step in the experiments, the question must arise in your mind that why Neurospora was selected for this experiment. The reason is that it can be easily cultivated, you can grow it easily on simple media. Its life cycle is short, which means that it rapidly grows into the next generation. Then its spore area is small. Also, the number of spores are limited. The spores are in linear sequence in a straight line. Then it has urea cycle like metabolic pathways, which can be studied for various purposes. And then we can study the very important one gene, one polypeptide, or one gene, one enzyme concept easily in Neurospora. Now, what did Beadle and Tatum do with the spores? They took the mutant spores that are treated with X-rays. Now these mutant spores are added in a complete medium. Now this complete medium has all the nutrients. Obviously the mutant spores, they grew perfectly in the complete medium. Then they took the spores and divided them into almost 11 test tubes. The test tubes have minimal medium. Then they added different kind of nutrients in each of the test tube. In the first one, they added pyridoxine. In the next, they added para-amino benzoic acid. Next, choline, then inositol, then nucleic acid, then folic acid, then niacin, riboflavin, thiamine, arginine, and the last one is the minimal control. The last one is minimal control. Now they wanted to observe that which nutrient was deficient for the growth of the mutant spores. Because of that reason, the mutant spores were unable to grow on the minimal medium. Now, what did they do? They observed that the test tube containing arginine, in which they added arginine, it showed growth. Now, what does that mean? That means that the enzyme which is responsible for synthesis of arginine was absent in the mutant strain. Why? Because we treated the strains with X-rays, and X-rays damaged the genes which encoded for this enzyme, and hence the synthesis of arginine was inhibited. And ultimately arginine could not be synthesized. It was absent inside the spores, and that is why they could not grow. Now, because of this experiment, it led to very famous one gene, one polypeptide concept. This is very important concept and it is our next topic of discussion, but you can see clearly the concept presented by previous scientists that X-rays, they damage the hereditary information. Because of that, the particular enzyme for which that gene was encoding was damaged, and hence the synthesis of arginine is stopped, and because of that arginine could not be synthesized inside the spores and their growth was inhibited, and hence they could not survive on the minimal medium. This resulted in the formation or generation of the concept of one gene, one polypeptide. Now let us see what is one gene, one polypeptide concept.